Good afternoon, everyone. Thanks very much for coming today. My name's Andrew Bate, and I'm the analytics team lead in the epidemiology team in safety at Pfizer, which means I lead a small team, looking at epidemiological and analytical methods for analysing observational data for the aim of discovering more and ensuring we know as much about the safety profile of medicinal products, and specifically Pfizer products, as possible. I'm also an adjunct associate professor of clinical pharmacology here and also affiliated with the CHIBI. In addition, I have a visiting professor appointment in the UK at Brunel University in the Information System Department there. So today, I'm very pleased to be with you. I'd like to talk to you a little bit about drug safety surveillance. So my intention is to give you an overview of the need for drug safety surveillance and talk about the methods and approaches that are currently used in order to try and discover and analyze side effects of medicinal products, with the aim really of stimulating or motivating that there's a need for ongoing research here, and there's lots of potential for interesting and exciting research in this real-world data, and I believe increasingly lots of funding and grant opportunities in this field. And then at the end, I'll make some comments about what, from my personal perspective, are the primary research directions and challenges moving forwards. This will necessarily be a personal perspective, and most of the results and examples I give will be based on research I've done both at Pfizer and prior to that when I worked for the World Health Organization in Europe. But hopefully that will give you a sense of the natures of the problems we're trying to analyze and some of the appropriate approaches, and also some of the clear limitations and the opportunity for future work. So I'm kind of going to try and get to the methods and results as fast as possible, but to give you some background, there is a need for ongoing monitoring of pharmaceutical drugs when on the market to ensure the detection of safety issues that unavoidably have not been detected prior to drug approval. This is because of wider use in real-world healthcare are the primary two reasons for this, so that there are certain side effects that obviously we do detailed analysis, many players involved, of medicinal products prior to their launch, primarily through clinical trials once they're into human use, but also previous testing and development in other toxicological examinations, investigations and clinical trials. The challenge is that there can be idiosyncratic adverse reactions that occur very, very rarely, that are very serious, that we need to know about. For example, certain types of very serious anaphylactic reactions, skin reactions, things like Stevens-Johnson syndrome, agranulocytosis. These sort of reactions, even if they only occur in 1 in 2,000 patients, because of the nature of their severity of things, we A, want to know about, and B, may, under certain circumstances, want to adjust how we use that medicinal product and which patients. So it's critically important, despite all the extensive and important analysis we do prior to a drug launch, that we continue to monitor these products once they're in routine use, and when needed, take further actions such as adjusting how the drug's used or the patients is used in. In terms of surveillance, there's a very specific use of a term signal that's common and will be a theme throughout this presentation. So just to be clear, I wanted to put that definition up for you right at the beginning. We talk about a signal of suspected causality, and this is defined within the field of post-marketing surveillance and drug safety in routine use, as information that arises from one or multiple sources, which, whether it's observations or experiments, which suggests a new potentially causal association or a new aspect of a known association between the intervention and an event or a set of related events, either adverse or beneficial, that is judged to be a sufficient likelihood to justify verificatory action. So some of the key points about this is that it's information of some source, that it arises from no specific data source. It's any potential data source. So this may be a medicinal product's on the market and there's a new clinical trial that data arises from. It may be that there's data in a certain electronic medical record database that suggests that there's an arising signal, and that there is some belief that there may be a causal association between medicinal product and the disease. But specifically, signal is used as potential causal association. Many of these data sets that I'm going to talk in more detail about now are they're real-world data. They're observational, they're noisy data sets. So there's an entire field dedicated to the identification of potential emerging relationships that then can be defined as clear hypotheses that are then evaluated further to determine, as far as possible, whether there is a genuine causal association. It can be either adverse or beneficial, but the intention then is to highlight issues that should be analysed further in some way. And this reference is, I would say, probably the standard in the field at this point, and it's taken from a recent publication by an organisation called CIOMS, and that's the name of the book at the bottom. Where possible, I've tried to be explicit on where references are coming from. If there's anything that's not clear, please do ask at the end of the talk. The primary way that we, as of today, and have done now for 40 or 50 years, discovered about new emerging potential side effects of drugs is through so-called spontaneous reporting. Are people familiar with what spontaneous reporting is? Could you just raise your hand if you know what spontaneous reporting is? So a few of you. So the idea is that in order to get a sense of what's happening in the real world, there is international and national systems whereby initially, when the systems were started up, clinicians, when they saw their patients, if they suspected that there was an untoward medical occurrence occurring that might have been due to a drug, they were urged to fill in a specific form outlining the rationale behind their belief and data, for example, what drugs were taken, date of the onset of the disease, other information, and asked to submit that report. And this was done, these systems were typically set up in the mid to late 60s, a huge increase primarily after the thalidomide disaster, and now we have these systems in, I would say, probably up to now 200 countries around the world, organized systems, both done from a pharmaceutical company perspective. Each company has obligations to collect these reports in order to discover as much as possible about their own drugs, but also regulatory authorities also have systems in place to collect these suspicions. And these reports are analysed locally in regional centres, for example, on a national level and also on an international level. So, for example, here in the US, the MedWatch reports is one of the systems that collects them, are all collected by the FDA, who has a database now of somewhere in the region of 4 to 5 million suspected adverse drug reaction reports, which they then analyze to try and detect new emerging problems. These databases are increasing in size exponentially, A, because it's easier to submit these reports through electronic media, also because collection has now been broadened to other healthcare professionals and also consumers. So, and I'll discuss this in more detail in a moment, but what's interesting about this data is it's highly sensitive in that potentially anybody anywhere in the world who has a suspected adverse reaction, that data can be collected. But there are also, it's very messy real-world data, so there are lots of challenges in terms of how the data is analysed. As of today, it is a valuable tool in the detection of previously unknown drug adverse reactions. I would go as far as to say it is the primary data source as of today in terms of determining a previously totally unexpected adverse drug reaction. And there are huge numbers of examples in the literature of now accepted causal relations that were first detected using spontaneous reporting systems. One of the challenges is that reports of adverse events are associated with a drug are not, of course, necessarily true adverse drug reactions. We have suspicions being reported. Well, why do we ask for suspicions to be reported? Well, the aim is to capture the things that are completely implausible, that there is no, at that time, mechanistic motivation, that just seem unlikely. Things like the antipsychotic drug clozapine with heart problems like myocarditis. Captopril, the ACE inhibitor, first time that coughing was detected, it was very unexpected. No one believed, or there was limited understanding and belief that this could genuinely be drug-related. That's now a well-accepted causal relation. So the idea is, well, let's try and lower the threshold of suspicion, ask people to submit things that they believe that there is something about the patient that makes them believe that the drug caused the adverse event, and then we analyze this data. And hypothesis generation about these sets of spontaneous reports then is referred to as signal detection. So, and I've alluded to some of these already, but just to summarize this type of data, and I'm going to talk about analysis methods for spontaneous reports at the beginning, and then I'm going to go on and talk about electronic medical records and some of the emerging research that's going on there. One of my reasons for starting with spontaneous reporting is that analysis has been done for a longer period of time, so that some of the problems and some of the methods, I think, that are being used on spontaneous reports are only now beginning to be used in electronic medical records, and I think it's interesting to compare and contrast the two in terms of what's possible and not possible. So back to spontaneous reporting limitations. Well, one of our problems is, because this is a voluntary systems, most commonly so, we have limited clinical information on the reports, and satisfactory secondary case evaluation is not always possible. So if someone says there's a hepatic type of adverse reaction, one would like a confirmation. Sometimes we get that confirmation, sometimes we don't. Similarly, if there's a certain type of skin reaction, we'd like to know if it really is a rash or is it something more specific like a Stevens-Johnson syndrome. Obviously, not all adverse drug reactions that are genuine adverse drug reactions will be recognized as drug-induced by a healthcare professional or a consumer for that matter. And even those that are suspected as being genuine adverse drug reactions will not necessarily be reported. So and then sometimes suspicion will mistakenly rest on the drug, and we get coincidental reports. And this is often the case if for large, widespread use of a product and common adverse events, diseases, where there's a coincidental near-temporal relationship. So, and this is one of the challenges of analysis in this field, so that if someone takes a medicinal product today and they have a disease tomorrow, one tends to believe that there's a link between the two unexpected occurrences. Some of those will be chance occurrences. So how do we unpick the chance occurrences from the general causal relationships? So we have a great deal of difficulty in terms of how much can we interpret numerically the number of observed occurrence of drug and event. We also have the problem about a lack of control information. We don't routinely collect the number of patients who got a certain disease without having a medicinal product, nor do we capture the total number of patients who took a medicinal product and didn't get an event. Now, there are some systems in place, but they're not collected within our spontaneous reporting systems. So here's the dilemma. We have this data set that's very large, that's very messy, that has been traditionally used for detecting adverse effects, and there is lots of evidence that it's very useful for that. And increasingly large numbers of this type of data, but there are fundamental limitations with it. So how do we develop quantitative methods and techniques to make the most of the strengths and minimize the impact of the limitations and weaknesses? So there is then an interest, as these data sets get larger and larger, to use quantitative methods to analyze these data sets. Where 40 years ago there were relatively small numbers of paper reports, clinical review of a case series by an expert in drug safety was done. That's no longer possible in many of these data sets because of the hundreds of thousands of reports that are submitted every quarterly, for example, in some data sets. So the aim then is to detect potential signals for further investigation that are not readily apparent at case entry. So if our quantitative methods just highlight cases, individual cases that are particularly likely to be plausible adverse reactions, well, it's not adding very much. What we're particularly interested in is pattern recognition, trying to find unexpected patterns that aren't obvious from any single report. The nature of these reports means that we have to be realistic. Clinical review of our outputs is still going to remain critical because of the messy nature of the data. our interest is, can we develop algorithms and methods that will focus our subset of interesting data amongst all the, as it has been termed, searching for the needles in the haystack. Can we reduce the area of the haystack, the volume of haystack that is most likely to represent the underlying needles? And nowadays there's been much work on this. There are several studies that showing there is reasonable predictive value of quantitative approaches. When I say reasonable, I would say positive predictive values are often estimated in the range of 40 to 50%. Negative predictive value, 90% or so. So far from perfect, but better than chance. And as I'm going to go on to say, these methods are now they're very basic and are used extensively, and there remains huge areas of research in how to better use this type of data. And just to reiterate, really, the nature of this type of data limits its role in pharmacovigilance, and we need complementary types of data, and particularly in observational data terms, the use of claims databases and electronic medical records is a primary area of research at this point. So that's I'm going to just skip quite quickly through this. The history of the quantitative methods started being used in the 60s and 70s, primarily using these types of spontaneous reports. Then there was the idea of surveillance in hospital-based systems, and a lot of work in the 70s in case control surveillance. And then in the 80s and 90s, people tried to link these reports to denominator data, whether it's sales data or prescription data, and look at reporting rates to try and highlight emerging issues. And then since the 90s, there's been a shift towards disproportionality screening. I'm going to skip over that. And then say that there are many new emerging methods, but all tend to focus on the same basic idea. So just to very quickly summarize them process-wise, and this is just one example of a process. The aim is that spontaneous report type data comes in to a spontaneous reporting data set. We quantitatively tag every single drug event combination in a certain database. And then based on some filtering algorithm, we have a subset that's generated in an automated manner, and we focus our clinical review on this subset. And of these, a subset will be signals that will be disseminated further. As of today, there are problems with all of the routine quantitative algorithms, so there are also qualitative filters that are also in place to ensure that we don't miss emerging signals. And just really to emphasize, in terms of state of art, the routine method is incredibly simple. I think it works well because of the nature of the messy data, but just to give you a sense of it, what we do is for every drug event combination in a given data set, estimate an expected count and an observed count and look at the ratio of the two. So for a given database, imagine we're considering a specific drug event combination such as coughing and captopril. We subdivide every report in the database in terms of the drug in the event listed, not taking into account any other variables on those reports. Into this 2x2 contingency table, we have in box A, reports of captopril with coughing. B, we have all other reports of captopril. C is all reports of coughing for any other drug. And then we have D is the rest of the database. So A plus B plus C plus D is the total number of reports. And then what we do is we estimate how many reports would we expect of captopril coughing in this data set, based on general reporting of the drug and the event, and we compare that to what we observed. So it's a very simple observed to expected ratio. And there is some more sophistication associated with that, which I'm not going to go into all the details of now, but I'm happy to discuss later or offline. One of the problems with this ratio, why is this ratio useful and interesting? Well, it deals with the problem that A will often be large purely because it's a common drug and a common event. So if we just look at the total number of reports, then we tend to see drugs that are very commonly used, statins for example, and we get events that are very commonly used, that occur very often, like rashes, and that we have large numbers of coincidental counts. This handles that. Our problem is as might be very obvious, is that A can be very low, but we sometimes have very low expected counts. So, for example, a medicinal product that was launched 3 days ago, we don't have any reports of it, or very few, 1 or 2 reports. We have a very rare adverse event. Left finger went, forefinger went purple, or whatever it is. If we have just one observed count and we have a tiny expected count, then our observed to expected ratio can be very highly inflated. We get very large values of that ratio. What does that lead to in practice? That leads to lots of false positive findings. So one of the questions is, can we actually model the observed to expected ratio in a slightly more smart way, sophisticated way? And one of the approaches then is to use Bayesian methodologies to shrink that observed to expected ratio. So a simple, for those of you involved and interested in Bayesian approaches, I'll just quick aside. So a simple assumption then is a prior assumption of independence between drug and event, and then we use that to weigh down we estimate a posterior distribution based on the data, which is very limited in this rare combination, and we combine that with our prior to get a posterior count. What does this do? Well, it shrinks our observed to expected ratio when we have very small amounts of data, reducing the number of false positives. And there are more sophisticated ways of doing that, and one of the debates at the moment is how complex a Bayesian prior does one need. And 2 approaches. One is you make an assumption and go more down a fully Bayesian approach and start with a non-informative prior of certain, based on subjective assumptions about how we expect and want our observed to expected ratio to behave. Another approach is to use the already accumulated data on other drug event combinations and to follow an empirical Bayesian approach. I'm going to try and get through the methods and give you some examples here. So one of these approaches then is to use a measure called the information component. Positive values mean we're seeing more reports than we'd expect. Negative values mean it's a logarithmic measure, so negative values mean we're seeing less reports than we'd expect. It's infrequently reported. In discovery terms, we search for positive values of this particular measure. We get this shrinkage or dampening effect when we have low observed counts where actually we wouldn't act anyway. And when we have lots of data, our prior assumptions have negligible impact. Okay, so let me stop there in terms of the theory and give you a sense of what this means in practice. Well, this is some work we did now about 15 years ago where we looked at the association or potential association between captopril and coughing and specifically wanted to retrospectively roll back a certain database, in this case the World Health Organization spontaneous report data set, and look at how this measure, this information component, changed over time. So in this case, we start with a value of 0 when the database started, or even when we started getting reporting of captopril in this database. This was our prior assumption, that in the absence of any data of relevance to this combination, we assume independence between drug and event. Then what we see is a dropping in the IC value as data began to accumulate. And as I say, this is a retrospective analysis plotted cumulatively. What does that represent? Well, that represents increased reporting in the drug with other diseases and reporting of the event with other drugs. So we're seeing lots of coughing reports coming in, lots of captopril reports coming in, but none of the combinations. And that seems, so that the 0 seems unexpectedly infrequent. We'd expect to start seeing some, at least coincidental reporting of the combination. So the IC value drops, and then what you see in the 3rd quarter, this is quarters on the x-axis, we start to see reporting of the combination, and the IC value increases and stabilizes towards a value of 4. Credibility, 95% credibility in intervals shown around the IC value. What's interesting, though, and this was one of the first examples we looked at, was that this combination was not known until 1986, and that even the first reports in the literature were not until about 1983. Now, there's a huge step between saying we highlight this for further analysis and actually saying, okay, this is an established causal association. but this was indicative of the potential use of quantitative screening algorithms for analysing this type of data. Just a contrasting example, digoxin rash, we don't see a positive value at all. Does that mean we don't have any reports of the combination? Absolutely not. In this particular database, there was quite a large number of reports of the combination, 30, 35. However, digitalis was a very commonly reported drug, and rash is, this particular database, the most commonly reported adverse event. So we would expect a large number of reports by coincidence in the database. So this would not be highlighted for clinical review, and indeed this was not something we would hope our quantitative algorithm highlights. Where are we now? Well, there are, we're still looking for clarity on best practices in signal detection. As I say, these very simple methods work reasonably well, but they're far from perfect. People are saying, is this the best data set to use on my particular organization? So there's lots of interest in developing new methods. There's still an ongoing discussion around, can we really not do any better? And we've done some recent research using, for example, Bayesian shrinkage regression analyses on these sort of drug event combinations to take into account, for example, co-reporting of several medicinal products on the same report. Challenge is, can we use more sophisticated methods and do better? Interestingly, so far, not really, except under clear specific scenarios where these methods are clearly limited. And then there's interest in more harmonization, understanding of how to really interpret these measures and what the next steps. One of the problems with these methods is that they're very focused on hierarchical terminology. So people, and I think this is such a clear area for better and more extensive research, and I know this is much more mature as a field in other areas of informatics. Most of drug safety is based on this idea that people are asked to report or classify using very specific hierarchical terminologies. So for example, in the terminology that they used in the WHO database, people were asked to record, is it a cardiomyopathy? If we're talking about a heart muscle disorder, is it a cardiomyopathy or is it myocarditis? So if you use a method that is based on the terminology and people are misclassifying myocarditis as cardiomyopathy, you may miss loads of cases. This is fine from a kind of an evaluation perspective in the sense that we, for a very clear, well-defined hypothesis, we can go and say, okay, well, I want to review all the cardiomyopathy cases as well and really determine whether they were all myocarditis. But in terms of hypothesis generation, knowledge discovery, terminology issues really limit our capability for analyzing and detecting issues early. Or at least potentially do to a large extent. And there is increased use of terminological reasoning, both in using a specific terminology, but also in how do we map between different terminologies used in different sets of data. Also, database term clustering of similar ADR terms is, I think, an area that there could be more research, and I'll give you an example of that in a moment. Here's a couple of examples, and really it's a quite sparse literature in terms of safety. One is trying to use ontological modeling of adverse drug reactions, another on using text mining, and in these reports we often get interesting information in the text listed. One of our challenges is that all of the reports have to be, and the medical records have to be anonymized, so how do we make the most of what we have without always being able to find out more information? And there's still limited use of text mining to facilitate anonymization of the case report narratives, and still the standard is to remove narratives when this data is made publicly available because of concerns about privacy. And I think that's a very fertile area of research. I'm going to just touch on it, but very limited in drug safety, linking of chemical structure to adverse event profiles. This is one example now, 20 odd years ago, using a neural network type application to look at prediction of different types of antidepressant drugs and looking at adverse reaction profiles. It was done on a very limited data set, and it's not even very well referenced, and I think this is an area that we're going to see more activity going forwards. Another major problem is we generally talk about detection of drugs and adverse events without going anywhere down the personalized medicine route in drug safety. So, for example, most of those analyses that are routinely used focus on drug and adverse event. And there can either be clear confounding due to other variables, or alternatively, stratum-specific effects. And this was something we published a couple of years ago where we made the point that you may miss things. And this is an example. I'm not saying anything about, this is just a theoretical example. Real-world data, but not going to get into whether it's a causal association or not. But it was noteworthy that if you look at the just the combination of reporting of loperamide and urinary retention in this particular data set, if you look at the crude observed to expected ratio and adjusted rates, you get a sense of no confounding going on. If you then subdivide the database by age and gender, just very crudely into children and adults, I think we did over and under 18, male, females, then we see very different reporting patterns in children and adults, which is totally missed as a potential effect modifier by looking at an adjusted estimate and just trying to ignore this variability. One of our challenges then in terms of stratum-specific searches for effects is that we very often run into small numbers. We end up with issues with the terminology and the way that we classify our different strata. So it's far from, I think, obvious how this would be done best, but again, another important area of research. I'm not going to go into the details of the method, but just as I talked about the need for syndromal type detection, one of our issues is, of course, that if a syndrome type or any disease occurs, then lots of other things are likely to happen at once. So can we do better analysis of these complex clusters of diseases that we would observe in the context of a data set where we're only getting some parts of the pattern reported? So we used a recurrent Bayesian neural network, again about a decade ago, to look for clusters and patterns in this type of data. I'm not going to, so just visually, and this is in fact training data that we used to test the method, to train and test the method before we looked at real world data. Imagine a world that we have 8 underlying syndromes, and there were only 81 possible different terms, diseases that could be sent in to us, so that the entire number of variables of different diseases is 81. And for each of these patterns, the red square represent that in that syndrome a disease is associated, and a white square represents that the disease is not associated with the syndrome. So we have 8 separate patterns. What do we get in terms of spontaneous reporting? Well, we get bits of each. People will see some of the symptoms, will ignore some of the symptoms, will add some symptoms that they thought were related that weren't. So what do we actually see? Well, we get data like this. We get samples of these underlying patterns. And then the question is, can we use pattern recognition, unsupervised pattern recognition, because we're again in signal detection here, to try and detect the underlying patterns? And this is the outputs with this particular algorithm we found. So could we learn based on incomplete things? I know this is very theoretical, so just some real-world data. When we ran it on the spontaneous reporting system, we looked at a drug that has a very well-known syndrome linked to it, the neuroleptic malignant syndrome, with lots of different types of symptom associated with it. And what we said is, okay, based on the reports that we have in the database, 9,000 in total on that drug, some of them will be associated with the neuroleptic malignant syndrome. Can we detect enough aspects to get a pattern of disease that would be indicative of the neuroleptic malignant syndrome? And this is one of the output patterns. We got some other output patterns associated with other attributes of haloperidol. And what this graph represents, and I know it's hard to take in, so just to get a sense of what's here, here we have the different hierarchical terms used in this particular dictionary, and they're listed both on the X as a column and a row, and then the numbers here are the number of co-occurrence of a pair of disease terms. So in this particular algorithm, we focused on pairwise occurrence of different disease terms, and using that same measure before, this information component, what the blue cell represents is a positive information component. So in this case, the number 6 means we have 6 reports where for haloperidol, these 2 disease terms were both co-recorded, and we didn't expect to see that many based on the number of reports where solely that disease was coded or solely that one was coded. And what you see from this pattern is that this is a constellation of a series of about 15 different terms that are unexpectedly co-reported. As you can see, there is no individual term that is co-recorded with every single other term, but what you see is that there's generally a large amount of blue here, meaning there's a large amount of dependence between these terms. And for the more clinically inclined, here are some of the terms that were highlighted. So this was done in unsupervised ones. We said spit out patterns that have some codependency going on, irrespective of what the terms are. And this one, after clinical review, was clearly linked to the NMS. So there's hypotonia, fever, tremor, confusion, CPK increased. I should add, though, that this algorithm worked less well on other examples. So again, an area where there is promise, but it's a challenging problem. I'm conscious that time's going, so this is the last example before EMR data. I mentioned anonymized data. This was a method we did looking at record linkage techniques, but rather than linking across databases, we focused on learning within an individual database. And one of our huge problems in terms of false discovery rates and prediction rates is that the same occurrence may be reported by different people, and we don't necessarily know that because they're anonymized, or alternatively, we may get follow-up versions of the same report that haven't been linked properly. So you get a belief that there's something underlying there that isn't truly. And what we used is a method based on a statistical approach from Hoppus and Hilton called a Hitmiss Model, and we developed it for use on spontaneous reports. And our problem here is that we wanted to get away from just a simple matching algorithm because reports with less data on are more likely to be identical. So if I have 2 reports that have no information apart from a commonly used drug and an event, then they will be identical. But do we think they're duplicates? Well, they might be, but we're not going to trust our judgment on that. There's too much indifference. So the question was, could we develop an algorithm that would, based on what that we can find informatively similar, so similar type technique to that used in, say, fraud research, can we find case reports that look unexpectedly similar? And this is an example of the type of thing. So we trained the algorithm on the data, all the data we had, to get a sense of what types of variables are very rarely similar. We adapted it to deal with both binary variables and real-world variables, like age. And then we ran it after some training to see what sorts of clusters of reports were similar. So for example, and this is real-world data then from that same database, 2 patients, 2 separate reports, are they duplicates or not? Age of the patients was different but very similar. Both females, both from the same country. 6 of the drugs listed were identical. 1 report had a 7th drug, the other didn't. The 3 adverse event terms were totally different. The onset date was very different, or seemingly very different, 10 days apart. Then you look at it in more detail and you just see there's just 1 single digit that's different. Outcome was undiagnosed. So it's a scoring algorithm that looks at each of these variables and says, are you the same or not, hit or miss? And then for certain combinations that are rare, it gets a higher weighting score. And that's the type example. That one was a duplicate. Here's an example that wasn't, that also I think is an area of interesting research and observational data. In this sort of data, we assume independence between all the different reports in terms of our routine signal detection algorithms. Look at this report here. 3 reports, all from the same country, same onset date, totally different ages of females. All had the same 1 drug listed and the same rare adverse event, and the combination was extremely rare. So looking at that data, you'd think, as the algorithm did, well, they're probably duplicates. There must be some error in terms of, or likely duplicates, definitely justify further evaluation. The ages are different, but that may be some data error in how they've come in. We should look at these reports. We went back to the individual reporter. This is when I was at the World Health Organization, and they were confirmed as non-duplicates, but they're all reported by the same dentist on the same day. So our statistical analysis and assumption of independent is clearly violated in this example. So what we were doing when I left the Dubbit show was using this algorithm to downweigh combinations of reports that were less likely to be independent and looking at how that influenced our ability to detect signals. Because obviously this type of finding can lead to large amounts of false positives. And I think this type of algorithm has got lots of other potential applications and other types of observational data. And in fact, I'm not sure I'll be able to go, but there's a censoring conference coming up in Washington, DC, and they've asked us to go and present this algorithm as a potential useful method for looking for issues and challenges in large censoring surveys. Electronic medical records. So this is a huge area of research at the moment, and this has been, in the US, driven by an act that was passed in 2007, the FDA Amendments Act, which called for access for the FDA to 25 million patient records by this year, and or last year, and 100 million patient records by 2012 should be available for analysis within the US. And at the same time, the FDA started what's known as a Sentinel initiative to test data models and explore methods for analyzing this type of data. So this is a huge, in terms of drug safety, major focus now with huge amounts of funding being poured into it. Now, to be clear, these observational databases have been collected and developed for many years. These are anonymized large databases that are routinely used for hypothesis testing. So we use them, for example, within Pfizer and other organizations all the time. They're licensed with many tens of millions of patients, but we use them routinely for hypothesis testing. So we have a very specific question in mind that we go and design a well-thought-through study to take into account confounding, what's the hypothesis that can be addressed appropriately in this type of data, and analyze them. The big question in the field at the moment is, can we do hypothesis generation? So you heard all about spontaneous reporting. We've kind of, yes, there is some methodological development, but there are fundamental limitations with the data that limit how far we can go. What's now being talked about is, can we usefully do signal detection and signal refinement type methods in this sort of data? So just to give you a sense of that again, and I'm focusing then on medicinal products once they've been launched. We're talking about the novel use of claims, so medical claims databases and electronic medical record databases for signal detection and refinement. As of today, it's routine to do signal evaluation with a predefined hypothesis where we have a subset of well-defined, plausible signals on the table. What's being explored now is, can we actually do signal detection? Less well-defined, more fuzzy signals, large-scale screening for finding emerging issues, which then we try and analyze a bit further before we get to the point of doing large-scale, expensive, time-consuming studies to really determine as much as we can. So just to give you a sense of this funnel, here on the far right, we have time-consuming studies. We're testing the anticipated or suspected. They're very convincing because we've considered issues like multiple testing. Here on the far left, we're talking about rapid, fast analysis, detecting the unexpected, and necessarily less persuasive because we haven't geared our analysis to a specific issue of interest. And in medicinal terms, of course, we have the problems of the wide variety of the nature of different diseases we're interested in, from latent long-term events like cancers to anaphylactic shock, rapid events, drugs, medicinal products, vaccines that are used totally differently in different groups of patients. And this is an area of emerging issue. And the FDA Sentinel, as of today, I think now has access to 78 million patients. And I'm not going to get into it in too much detail, but the approach they're using is interesting in itself, at least in this field is new. because of anonymization, privacy regulations, rather than trying to share and centralize the data, they're focusing on a distributed network approach to analysis. So developing a common data model, analyzing, developing algorithms and methods for use remotely, and sending them through firewalls to run on different common databases converted into a certain common data model. I want to describe some work we've done on a particular UK primary care database that was used while I was at the WHO. This was, compared to some of the databases, relatively small, 2.5 million patients at the time of analysis, 113 million individual prescriptions listed, but compared to spontaneous reports, a much richer data set. We had test results, hospital referrals, admissions, all of the other fascinating and rich data we get in an electronic medical record. Obviously, this is observational data, so that there will be missing data, noisy data, and all of those issues, but still from a discovery, a data analysis perspective, lots of potential. So some key concepts prior to analysis of spontaneous reports, which is where we developed our particular methods from. Well, now instead of just looking at what happened in terms of suspected adverse reactions after drug use, we're able to look at what happened prior to drug use. We have diagnosis and tests. We have genuine drug utilization, so that there are serious strengths of this data compared to spontaneous reports, although there are clear challenges. The risk of false positive findings, because we have no clinical suspicion of causality in this type of data. But it's a very interesting data set to analyze. I'm not going to go through this in detail in the interest of time, just to say, right now this is such an emerging issue. A range of different types of methods are being approached, coming from different communities, from traditional EPI methods to traditional data mining methods, from methods that are more commonly used in RCT research and other methods. So it's a very interesting area. I'll come back to that slide. I'm going to just talk about this one. This is, I showed you the example on spontaneous reports. This is an example then on that IMS database where we looked at the IC value and adapted it to look at drug event combinations in medical records, adjusted for how often events occurred prior to drug use, and looked at retrospectively whether we'd highlight a known association early or not. And just as before, we're searching, we're mining for positive IC values in this particular application. Here you see retrospectively that the IC again starts from 0 because of the Bayesian implementation. It became quantitative in 2000, and that is prior to the labeling change for this particular combination. Here on the graph below, you see some more data behind this top graph, again lined up horizontally. The block graph shows the observed count. So by the end of 2005, there were about 36 times that edema had been listed in a medical record within a month of, in this case, a month of terbinafine use, prescription at least. And then the line is our expected count. So same kind of core principle of observed to expected ratio. Very few examples as of today about the potential use of this sort of data for signal detection. Here's another one. This was done at Pfizer in collaboration with a group in Harvard, looking at analysis of a distributed network of claims data, the HMO network it's called. And you see the same sort of graph, a retrospective evaluation, in this case looking again at angioedema, but this time for the ACE inhibitor lisinopril. And what you see here is the red graph is the one to focus on, and they, in this collaboration, just used a simple relative risk estimate. But again, comparing observed to expected, what you see is a sudden peak very soon after surveillance of this particular combination. Why did that happen? Well, pink is the expected count, blue is the observed count. We see kind of a constant increase in the number of times angioedema was listed in medical claims after lisinopril prescription, but this was very rare, this combination, this is very rare drug use, drug prescription use early on. so that a very small number of cases caused the peak, so that there is emerging evidence that there might be value in signal detection. However, even in big databases with 20 million patients, consider a combination where we have 1 in 200 patients get a specific drug within a year, which is a large amount of drug use. That's 100,000 patients. Of those, if 1 in 2,000 get a specific AE, which would be something we would want to detect, we have 50 events, which seems quite a large number. But in a medical record or a claims database, we may not get all events recorded, as well as selective recording or under-recording. We may not have events accurately or consistently coded, and back to the previous problem, if we're focused on a specific terminology, if people use the terminology differently, it may be hard to capture all our cases. There may be missing key clinical information behind those cases. And what about if we're talking about a prevalent non-drug disease, so where we're having lots of coincidental reporting of this or a recording of this particular disease? So patient records won't provide a panacea for signal detection. So what we need to move towards is signal detection or use in emerging data sets, but also traditional database. Plus signal refinement and further understanding of emerging signals may be where, at least in the short term, these particular data sets are most valuable. So just to reiterate, where could we do signal refinement? Say for a signal coming out of a clinical trial or a spontaneous report, we could quickly do further analyses using these sorts of data potentially to take into account time-stamped diagnoses, the exposure, look in more detail at the history of disease in these patients, and try and take into account in our algorithms the other data. And again, reiterating, observational data is not perfect. Just to give you a sense of then the types of things, yes, we might be able to find new adverse drug reactions, but new information on known adverse drug reactions, for example, unexpected time to onset type patterns, stratum-specific variations, effect modification effects, underlying disease, periodic patterns, drug-drug interactions. So there's a whole area of increased knowledge about drugs that we may not know everything about these types of issues or won't be able to before a drug's launched. In real-world drug use, can we discover more? Here's an example then of this type of thing. Acute pancreatitis recording relative to a omeprazole prescription. Again, this is the medical record database I mentioned before. But instead of time on the x, we've got IC value again on the y-axis. But instead of time in terms of time of the database, this is time relative to date of prescription of omeprazole. So what we've taken is every single patient in this 2.5 million medical record and lined them up by the date at which they received a given drug, in this case omeprazole, and looked at the recording of pancreatitis in monthly intervals, whether it occurred after or prior to omeprazole use. So here that first point is looking at the occurrence in the medical record of acute pancreatitis, irrespective of whether there was a suspicion associated with a drug or not, relative to the time of omeprazole use in this case. How many times did and if we start with the bottom graph, this block graph says that there are about 40 patients in this particular database who had acute pancreatitis listed as occurring in the month after omeprazole was prescribed for the first time. No clinical suspicion whatsoever. And what you see here, looking at this bottom graph in terms of observations, is we have a large peak around the time of drug prescription. Well, that's no surprise because we have more data. Generally, we have a censoring effect. We have much more data around the time of drug prescription because patients are going to the doctor for some reason there, whereas we're less likely to know what's going on if you go 3 years out, before or afterwards. So we see a peak around the time of drug use. Then, just as before, here's an expected count, and each of these refers then to the counts in a given month window. And what you see in the above graph then is the IC value. So it's the observed divided by the expected for a specific time. And what you see here is a much flatter graph compared to below. Why? Because we're kind of normalizing for the fact we have more data around drug prescription. And what you see here is a positive IC value. to the right, on the right-hand side of the graph. What does that mean? Well, that means that patients who had a omeprazole prescription are more likely to have acute pancreatitis in the months after that drug use, which might be indicative of an emerging problem, right? But what you see from this particular graph is that there is a large amount of positive values prior to drug use. What does this mean? Well, this means that patients who will go on to get an omeprazole in the future are more likely to have acute pancreatitis, which could be and is clinically very plausible based on perhaps misdiagnosed. The reason for omeprazole use might then be, in many patients, acute pancreatitis or underlying symptoms associated with acute pancreatitis. So there's a clinical link between the disease and the drug. And the interesting thing compared to then spontaneous reporting is that what we would see with spontaneous reports is we would see some large number of cases occurring after drug use, but have no idea of knowing what to compare that to. And so this gives an indication, doesn't prove causally, but it gives an indication that there's at least plausibility of confounding going on here that we can then analyse further. So the potential for this type of method is to be able to do this fast, to get a sense of explanations behind a finding, which then could be done in a much more extensive study that could take months to assess further. But we're able to do this sort of calculation and produce this combination for any drug event combination within a matter of minutes. So it's the idea of increasing the knowledge and understanding about a drug event combination very fast in order to better define our hypothesis further investigation. So to wrap up, there are, at the moment, so much needs to be done in terms of potential, at least, as a consequence of this FDA Amendments Act here in the US and others. There are several initiatives to better use this type of data. We're very good at discovering adverse drug reactions and determining whether they are causal associations and taking appropriate decisions. But there is a potential for doing even more using these types of data in different ways. So there are several international initiatives to determine whether we can actually see performance improvement, and these are just some examples of them. Some of them very heavily funded. And I just want to mention one of them before I conclude, and that's the OMOP, the Observational Medical Outcomes Partnership, which is open to anyone to be involved, which was initially a 2-year initiative. It's now been extended. It's a public-private partnership with the aim of improving the monitoring of drugs. And what they're doing is analysing healthcare databases from a methodological perspective and linking them together. So the idea is to take different commercial databases and to analyse them centrally, but also to work with partners to ask them to convert their databases into common data models and then to analyse them remotely. So they've developed a series of algorithms, I think 20 or so, which run on a certain database structure, a certain common data model, and then their collaborators have been asked to convert their databases into the common data model. And then voluntary participation beyond that. And they're doing a large-scale testing of methods to try and determine which methods work best, for what sort of drug events, and is this, how promising are these types of methods and approaches in real-world electronic medical records and databases, or is it something that's potentially very interesting but in fact brings limited value to the table? So what do? Well, different data sources converted to a standard format, running standard methods, producing standard outputs, and they are beginning to report out the results for some of these studies. And just to give you an example of some preliminary results that I presented at a conference on behalf of the group, a medical claims database, a small one of 1.5 million lives, pharmacy dispensing with diagnosis from both inpatient and outpatient services. They're focusing their analysis on known and established drug event combinations, and here you see the scores from these different combinations on that particular database, and this is just preliminary results. But you see the measure I mentioned before, the information component, and another measure. And what you see here then is, and I'm not going to go into all detail, is strong concordance between these two measures, which is not very surprising given that they're very similar measures. But what you also see is in this upper right quadrant is positive values, meaning things that would be highlighted for further analysis. You see the majority of these known effects stood out quantitatively first for both methods. So again, the finding so far is that there's some promise in identifying issues across this large amount of data. However, there's a major problem with a number of false positives still being highlighted. So these are the research challenges that, in my opinion, are facing the field. We need to develop methods with sufficiently strong performance characteristics, and that hasn't been done yet. There needs to be, and there isn't, there's a lack of agreement in the field on how to define and demonstrate appropriate performance levels. So what is a good operating characteristic of an algorithm? There is some work in linking terminologies. Many of these different, certainly internationally, many of these databases are using different terminologies. SNOMED, MEDRA, ReadCode, huge numbers of different terminologies. So developments for techniques for both clustering related diagnoses, getting beyond rigid classifications, hierarchical terminologies, using, for example, compositional terminology type thinking, but also mapping between the different terminologies. And also, at the moment, limited work on semi-automated manner of quantitative analysis to take into account other aspects, for example, free text information on EMRs. So to conclude then, will active surveillance methods be increasingly examined on this type of data? Yes, absolutely. Will they replace spontaneous reports for signal detection in the near future? No. I think, at least initially, they're going to particularly facilitate rapid signal refinement, so understanding more about emerging signals, and better understanding the need for and helping in study design of formal epidemiological studies, rather than replacing, because of the nature of the way these algorithms run, multiple testing and other aspects. And there are several initiatives investigating these emerging roles. Here are some other examples of related methodological development in analysis of active surveillance type approaches at Pfizer. As I say, we're moving towards, I believe, a toolkit for safety surveillance using clinical trial data and all these other types of data sets for active surveillance and safety surveillance. But formal studies will continue and will perhaps will, in my opinion, play an increasingly critical role for hypothesis testing of these safety issues. We won't get away from the need for formal testing of hypotheses. And we're moving towards, I believe, the need for systematic approaches that take into account use many different algorithms focusing different aspects to allow us to do discovery around this real-world data. And this is just an example of a prototype software published a year or so ago. There are the main references I've mentioned in this presentation. And just then to conclude, quantitative approaches add value to signal detection. Pharmacoepi studies for hypothesis evaluation do have this ongoing critical role, but there is some emerging evidence that surveillance of observational databases can contribute to the process of signal detection and refinement, but there is a huge challenge remaining of how to separate potential true emerging issues from the vast number of potential false positives that could emerge. As well as, this is very research focused, there's a huge amount of work that's needed to do before we can go from research into routine use. It'll take many years, I think, to get there. But then in the context of an academic perspective, I think there's a huge amount of exciting research potential in drug safety surveillance using these different data sets and combining these different data sets with an important potential practical utility. And I think that there is, and there has been a huge explosion of availability of data and also funding opportunities for this sort of data. Thank you very much for your time.